So, welcome guys. Today's topic is your Operon. And as you can see here written, that's a group of bacterial structural genes that are transcribed together. And all those genes are transcribed together, not all, but specific some cluster of genes that transcribe together by a control, and that to be controlled by a single promoter. That is what we call our Operon system. An important thing is that particular set of group of genes, that is your structural genes, have a operator region and its own promoter region. And this structural gene, along with its operator and promoter, are called operon. And this operon is controlled by another set of small gene that is called the regulator gene. And the most interesting thing is that this secondary gene has also a promoter. So once it gets activated, it will produce the proteins. That's what we call the regulator protein. When it goes and binds to that of your operator region, it will block the movement of this RNA polymerase from the promoter regions to the structural genes, and it won't be able to transcribe the protein of interest, and that's why the promoter will not be formed. So this is the thing. The operon is nothing but the structural gene with its operator and a promoter region, and it's controlled by another set of gene, what we call the regulator gene, having its own promoter. The regulator gene is not a part of Operon system. Okay, so this is all about this Operon basic concepts. One more thing also, you can see here that some space between the promoter and operator and your gene A, this little bit merged together. This because you can see here written the regulator gene is can be bind usually overlaps at the 3' end of the promoter and sometimes to the 5' end of this first structural gene. And in this shown this picture, you can see it's little bit overlapped in between those genes. So this is the Operon system.